I'm just coming back, right? In 1 minute, just printing something out.
Dans la technique, Mathieu, tu pourras éventuellement, pour la présentation des membres du jury, nous les mettre en un peu plus large ou pas? On a forcément le fond. Là, c'est intervenant, est-ce que tu es en galerie aussi? Voilà. C'est toi qui annonce, on commencera par une présentation. Ouais, tu peux commencer par toi, mais...
Tu veux se présenter même? Tu veux se présenter même?
Non, non, tu peux laisser se présenter.
D'accord.
Ce qu'on fait les dernières fois, on va laisser chacun.
Ça veut dire plein de parties, oui.
Oui, c'est ça, comme ça, t'as pas. C'est moins d'efforts et moins de stress.
Une partie là.
Ouais. On a perdu un membre du jury.
Ok, on va peut-être pouvoir commencer du coup. Donc, bienvenue à tous pour le thèse de Mélanie DEBELGARRIC. Bien, comme ça que ça se pense. Donc, je suis Louis-Miguel Chevin, directeur de recherche au CNRS au CEF à Montpellier. J'ai été désigné président du jury et donc je vais vous présenter maintenant les membres, ou je vais demander aux membres du jury de se présenter. So first we have Dina. Can you introduce yourself, please? Dina.
Hello, I'm Dina Lika. I'm a professor at the University of Crete in the Department of Biology, and my expertise is in modeling biological systems and particularly using the dynamic energy budget theory.
Voilà, donc Dina is the first opponent, rapporteur. Moi-même, je suis le deuxième rapporteur, donc je fais beaucoup de modélisation en biologie évolutive, génétique et écologie évolutive, génétique quantitative, et d'autres questions. Je passe le détail. So next, we have
So I'm Gonçalo Marques, and I'm a researcher at University of Lisbon, also working with the modeling with the dynamic energy budget.
Ben Bastien.
Oui, merci. In French, or in English, I don't know. So my name is Bastien Sadoul. I'm an associate professor at l'Institut Agro. I'm mostly a physiologist of stress in aquatic species, and but I'm also using dynamic energy budget models to link the physiological mechanisms and the fitness of the fish.
Okay, and then we're going to finish with the supervisors. So
very quickly for two people might not know us. So I'm Charlotte Recapé, so I was the main supervisor for the Mélanie thesis, and I am an assistant professor, so je suis maître de conférence à l'université Pompéi de l'Adour ici.
Et je suis enseignant-chercheur aussi à l'université de Pau et des Pays de l'Adour, et je suis sur la direction de thèse pour des raisons purement administratives.
So as usual, we'll start by a 45 minutes, something presentation by Mélanie, which will be followed by our questions.
C'est pour Rama avec qui on va partager, parce que sinon ils vont avoir droit à deux diapos à la fois. On va gâcher un peu le suspense. Diaporama PowerPoint.
So, good afternoon, everyone. I'm Mélanie DEBELGARRIC, and today I will present you the 3 years of my PhD, which is unraveling the evolutionary potential of freshwater organisms facing climate change through dynamic energy budgets. OK.
C'est l'effet zoom.
Ça commence super bien.
Fais-le avec la flèche, peut-être la première fois sur l'ordinateur. Des fois, avec Zoom, ça fait ça.
Vérifie que tu es sur la bonne fenêtre, simplement aussi.
Même sur l'ordi, ça ne marche pas.
Non,
Faut être dans-
tu ne vas pas être sur la fenêtre comme vous avez dû changer.
Voilà. OK. So sorry for that. Let's start with some introduction and context. So we all know that the genotype and the environment act together in organism to define a certain fitness through plasticity or other processes. But to link the fitness and the genotypes and the environment, we need to understand the traits and to quantify these traits. And in my thesis, I distinguished 2 types of traits, which is the phenotypic traits, like the growth, the weight, the length, the metabolism, or the life story traits, like the age of puberty, lifespan, reproduction. So if we talk about the traits, like phenotypic traits can be easily measurable on the field, like, for example, the length, the weight, which is used, for example, to estimate some growth rate or something like this. But also the respiration rate can be used to quantify the metabolism thanks to the metabolic rate. And at the other part, the life history traits are more related to the life cycle of the individual. So, for example, on the field, the age of first reproduction can be like used to estimate the age of puberty, the age of death for the lifespan, and moreover, the X production can be used to quantify a reproduction rate. So yes, the traits links these 2 processes, but how to link traits to evaluate the fitness? And for that, I use a theory, which is the pace of life syndrome theory. And actually, it can have a clue on the individual life history. By the covariation of these traits, we can define a certain strategy. And these strategies in organism can be fast. So it's the live fast, die young. There is a rapid growth and an earlier puberty, but the lifespan is reduced. And thus, the individual will reproduce earlier. And in another part, there is the slow strategy when the individual will has a slow growth and a longer lifespan, but it will reproduce later. So thanks to that, we can determine the fitness of organism. Moreover, there is an environment which can act as a selective pressure on the fitness, and this fitness can also alter some genotype expression. And it is known that in terms of temperature, for example, the stable environment will favor slow strategy, and the opposite, like the unstable environment, will favor fast strategy. But it remains here a gap, which is represented by this dotted arrow. It's how to link genotype and environment to the expression of traits. And for that, we need to quantify the bioenergetic processes, so the quantity of energy which is required to express the traits. Because with bioenergetic processes, it can make an evidence of the trade-off between growth, maintenance, and reproduction. For example, the individual will not have enough of energy to do all these processes, like the growth, the maintenance, and the reproduction. So it can impact the individual life history and thus the fitness of organism. Moreover, there is a physiological constraint which exists in the bioenergetic processes and with the environment, and which can act on the fitness. So this determines the first axis of this presentation, which is determining how variations in physiological processes could lead to different individual life history through the expression of the traits. And in the second time in this presentation, I will respond to how does temperature variation modify the fitness landscape for bioenergetic traits. So the first step in my methods, it's the bioenergetic modeling. So how to quantify the bioenergetic processes here. So it's for that I use the dynamic energy budget model. So just easily, like imagine an organism, it will assimilate energy from food sources, and this energy will be stored in the reserve, which can be, for example, the fat of the organism or something. And after that, the energy is mobilized through two physiological processes, which is in one part, the structure. So it can lead to the physical development, the length, the growth. And in another part, it's going into the maturity and reproduction, for example, to produce eggs. And in each part of the process, there is like some maintenance, so the somatic maintenance and the maturity maintenance, which is dissipated energy during the process. So the first one, the assimilation, if we look at the equation right now, so yeah, the F here represents the food sources and the V represents the structure. And here you can see a parameter, and it is called a primary parameter. So this one is my first of interest. After that, the mobilization flow. So it's a little bit more complicated, but it depends on the reserve and the structure, of course, but also on other primary parameters. So the volume specific cost of structure, the energy conductance, and the energy allocation to soma. Kappa, the energy allocation to soma, is actually the fraction of energy which going into the structure. And thus, the reserve, which is like a state variable, will be easily like the ingoing flux of the assimilation and the outgoing flux of the mobilization. After that, if the energy go with the fraction kappa here, energy will be dissipated by somatic maintenance. And here, the somatic maintenance depends on the structure, but also on another primary parameter, which is the volume-specific somatic maintenance rate. Finally, the energy will be stuck in the structure. And here we can see that the structure really depends, again, on these two primary parameters in color, in green, and in orange. In another part, there is also energy dissipated for the maturity and reproduction here. And there is another primary parameter who appears, which is the maturity maintenance rate. And finally, this is the equation to estimate the quantity of energy in maturity and reproduction pools. So actually, the individual when it is like not age-old, all the energy will go on maturity, and when this maturity age reaches a certain threshold, it will switch into maturity, into the reproduction here. So as you can see, there is the selected primary parameters, which has like a different influence on this Deb theory diagram. And for me, these primary parameters can represent the genetic aspect of the model, because each organism can have a difference in the value of the different primary parameter here. And so it could represent an inter-individual variation across a population. So this parameter, where it come from? Like it's come from a database which is Add My Pet. So there are observable traits here, and from these observable traits, there is an estimation, and after we have the estimation of the primary parameter, and thus with that, we are able to calculate different traits theoretically. And after that, the traits are compared to observable data and see if the model match or not. So here, a gap remains is like how to calculate the different traits here, like the physical development, the X production, etc. So it brings me to the second step of my method, which is the quantification of traits from theoretical states variable. So here, we are here in the diagram, quantification of traits. So the first one is the specific growth rate. So the specific growth rate depends on the von Bertalanffy growth rate here, and you can see here the different primary parameters that are exposed earlier. And here it's the ultimate length. So it's like the maximum length the organism will reach during its whole life. And here you can see also the primary parameter, kappa, PIM, and PM. So for the specific growth rate, it will depend on this part of the diagram. For the weight at puberty, it will depend on the reserve and also on the structure of V here, so here in the diagram. For the mass-specific respiration rate, it will depend here on the reserve structure, but also the somatic maintenance. For the lifespan, which is firstly the mean age at death, it will depend on the survival of the organism, but also on two primary parameters, the somatic maintenance and the energy cost of structure. So the primary parameter, which is related to growth here, so it will depend on here in the diagram. For the age at puberty, it's easily like the time when the maturity pulse is reached and when the individual will switch to puberty from adult stages. And finally, the reproduction rate is like the eggs production, the number of eggs produced. So it will depend on the structure of the organism, but also of the energy which is in the reproduction pool. So it brings me to the first part of this thesis, which is traveling bioenergetic parameters and traits. So this in my diagram. So for some initialization and context, for this first part, I will use Daphnia magna as a model species, and the individual will evolve in a constant and optimal temperature environment and with ad libitum food source. And thus, thanks to the DEB model, I can, from this DEB primary parameter, estimate the different traits. But this time, I put some parameter variation in the DEB primary parameters following the range of possibilities to run the model. And each parameter will vary one at a time. So as the parameter vary following the range of possibilities, it can be possible that some values are inconsistent with nature. And so, as you can guess, like each parameter has its own influence on the bioenergetic distribution of the organism. So for example, for the specific assimilation, it will increase energy input towards all the functions. So in structure, in growth, but also on reproduction and the X production. In another part, the energy allocation will increase the energy shift toward growth. So individual will be like more, with more weight, more tall, but they will reproduce less. After that, the somatic maintenance, the energy directed to the maintenance of the soma, which can lead to like a reduced lifespan, because if the somatic maintenance is really high, there is a lot of energy used to repair the damage, the cellular damage. In the same way, like when maturity maintenance increases, there is an increased energy required for the maintenance. after the cost of structure increase energy to maintain the structure, but the difference with the somatic maintenance is that here there is a lot of energy will be taken in the reserve, so the individual will have less reserve than with somatic maintenance. Finally, the energy conductance increase energy toward growth and reproduction, and that's the same way as the cost of structure. It will take a lot of energy in the reserve. So now I'm looking for the traits who are quantified thanks to the DEB model. And this is the kind of results that I can have thanks to the simulations. So here it is the example of the lifespan. And you can see that it can be positive correlation. So here for the energetic cost, the energy allocation, and also the specific assimilation, the curves are positive. It can be also negative correlation here, like for the energy conductance and the somatic maintenance, or we could have like no correlation, like here with the maturity maintenance. And so I did this kind of graph with all the parameters and all the traits here. And my main question is, does the interaction follow the pace of life syndrome hypothesis? So just for remembering, fast strategy should have rapid growth leading to high metabolic rate, but shorter lifespan with early puberty. And slow strategy is a slow growth and a slow metabolic rate with a higher lifespan and later puberty. So with this question, we can see that for assimilation, it does not follow the pace of life syndrome theory. But in the contrary, for the energy allocated to kappa, like it could be apparented to a slow strategy. I forgot to tell that this is the result when the parameters are increasing, okay? So when kappa is really high, it could be apparent to a slow strategy in terms of pace of life syndrome theory. After that, for somatic maintenance, it's not the case because the growth rate is like with a negative correlation, and the age at puberty has a positive correlation, which is not expected when we have a reduced lifespan. Okay. Here, there is no correlation with the growth rate and the lifespan. So actually, the maturity maintenance is not a good indicator to see if its variation can interact with the pulse hypothesis. The energy cost of structure does not follow this theory either, but the energy conductance, when it's increased, it could be related to a fast strategy, like here with like a growth rate which is really high and a lifespan which is reduced and earlier puberty. So in conclusion of this very first study, like we can see that energy allocation and energy conductance can, as a variation of these parameters, can be an interpretation of pace of life syndrome theory with a high energy allocation leading to a slow strategy and a high energy conductance leading to a fast strategy. But for the other parameters here, there is no clear strategy. So the parameters independently do not define a specific strategy in organism. So in the second part of the thesis, I will predict fitness landscape in a changing environment. So in this time, I will take into account the environment and the temperature, but also I will speak about fitness. So for some context and initialization, in the first part, I will study Daphnia magna, and each individual are in a constant temperature, but different temperature between individuals. And in the same way, I have the primary parameters. I will quantify the traits, and I will quantify some response to the environment. In the second part, I use Salmo truth, so the brown truth, And the ground truth is confronted to difference in amplitude of temperature during its own life. So there is, in a part, low amplitude here around a mean temperature, and a high amplitude around this same mean temperature, which is almost 8 degrees. And after this kind of simulation, I will study the influence of temperature on the response, but also the influence of temperature and the primary parameters on this response. So this response, I chose to study the lifespan, the lifetime reproductive success, LRS, and the intrinsic rate of increase, R prime. So how to quantify the selective values? So the first one is the lifespan, which is the survival curve. And in these simulations, it's when the survival is like really, really low, the individual die. After that, the lifetime reproductive success here depends of the R, which is the reproductive output, which is like the X production of the individual, and it depends of the survival. And actually, it represents the total number of offspring an individual produces over the course of its lifetime. And for me, it captures the realized ability of an individual to contribute genes to the next generation. And finally, thanks to the lifespan, the lifetime reproductive success, I can calculate the intrinsic rate of increase. So with here, the Neperian logarithm of the LRS and the generation time T here, which is the average time between 2 consecutive generations in the lineage of a population. So here, the intrinsic rate of increase is interesting because it can be a proxy of the individual fitness with a higher R prime, which indicates a fast strategy favoring early reproduction and higher offspring production, and a lower R prime, which corresponds to the slower strategy with delayed reproduction and fewer offspring. After, I have to model temperature, and for Daphnia magna, it's a constant temperature for the lifetime. So it's just a temperature corrector, which is applied at a certain point of the model. And I just change the ambient temperature here between the different individuals. And for Salmotruta, there is an amplitude variation, and it's basically a sinusoidal curve around the mean temperature. So the only parameter here which will change is this A, which represents the amplitude of variation. So in the first round, I will present you the results here, so the influence of temperature. So in the simulation, there is like 100 individuals for 100 days for Daphnia magna and almost 20 years for Salmotruta. And the value of each individual are like the value of estimated in AdMyPatch database. So you can see here that in all cases, the intrinsic rate of increase with temperature, while the lifespan decrease with temperature. And for the LRS here, you can see that there is high egg production with temperature for Daphnia magna at constant temperature, but not for Salmotruta when there is like a difference in temperature amplitude. So it matched with this hypothesis, which is author is better because the intrinsic rate of increase, which is a proxy of fitness, increase with temperature. But a question remains, it's what happens if primary parameters vary? So for that, I will just focus this presentation on the result of Salmotruta, so with variation of temperature amplitude. And here, this is the kind of results that I can have. So here, there is the difference of temperature amplitude, and here the different values of the different primary parameters. And thus, the colors represent the value of the intrinsic rate of increase R prime. So you can see different profiles. For example, the black triangles markers here are the best fitness for each values of parameters. So here, for example, for the specific assimilation, the fitness is better when the specific assimilation is really low. For the energy allocation, it's the same. It's when the value is low. And also for the energetic cost. But for energy conductance and somatic maintenance, the fitness is better when the value is high. And there is no best value for the maturity maintenance. And in all of the cases, you can see that higher amplitude increases the response, like in all cases, because here we can see the highest value of R prime. So with this first graph, I can determine a certain profile, like the fitness is better when specific assimilation, energy allocation, and energetic costs are low, and the fitness is better when somatic maintenance and energy conductance are high. But maybe there are some limits, because parameters are varying one at a time. So could there be any changes if parameters covary together? So I determined some profiles, which is not changes in fitness landscape when parameters still have the same influence together. Like, for example, if individual has low value of specific assimilation and low value of energy allocation, the fitness will be still the better. Or, for example, if there is low values of PIM and high values of PM, this combination can be favored. But here, it can seem counterintuitive because this combination leads to low reserves and high maintenance, which would reduce the lifespan, so maybe the fitness. So maybe it can, we can have like changes in fitness landscape with factors acting together to create an additive or compensatory effect. So just for remembering, the fitness is better when energy allocation is low and somatic maintenance is high. And thus, I varied the parameters 2 by 2. So here there is the value of somatic maintenance and here of energy allocation. And there is the value here of the intrinsic rate of increase, but also the difference amplitude. So no amplitude, 5 degrees of difference in amplitude and 10 degrees of difference in amplitude. And you can see here that for a low value of energy allocation and a high value of somatic maintenance, the best fitness should be here. And this is the case. So the best fitness in the combination is the best value of the parameters taken independently. And secondly, we can see an increase in amplitude leads to a stronger response, because here you can see that the fitness is much better when the amplitude is really high. But change in fitness landscape can occur. So here, the best fitness is at low values of specific assimilation, while the best fitness occur at high values of energy conductance. And when we covariate the parameters together, like a low value of PIM and a high value of V dot should be, but the best fitness should be here, there. But you can see that there is a lag, and for energy conductance, the best fitness is at this value of energy conductance. So there is a shift in the organism response, and an average value of energy conductance is favored when specific assimilation is low. And moreover, the response is still stronger at higher temperature, as you can see here, for 10 degrees of amplitude. So I did that for all the parameters varying 2 by 2. And we can see that here, when it's green, there is changes in the fitness landscape, which is not the case for this one. So in conclusion of this second part, like an increase of temperature amplitude leads to a stronger response in terms of fitness. And this highlights the importance of seasonality among freshwater organisms. And some parameters can covariate, leading to changes in the fitness landscape. But we need a quantitative study to unravel if there is additive effect or an interaction, for example. Like, for example, if there is like a parameter 1 and 2, and they're covered together, maybe an additive effect can occur, and here the fitness will be just the addition of the two parameters. But an interaction can occur if the parameters 1 has this influence, and the parameter 2 has this influence. Maybe the covariation of these two parameters can have this influence, which is not an addition, but an interaction. So now let's talk about the discussion and conclusion of this thesis. So the first part was to linking primary parameter to the expression of traits. And that theory has made possible to capture complex physiological feedbacks and may explain the inter-individual variability in population. And we saw that several profiles emerge. So a single physiological processes can significantly alter the expression of traits. It was the case of the energy allocation and the energy conductance, which allow individually to determine a strategic profile with fast strategy when energy conductance is high and slow strategy when energy allocation is low. Moreover, there is couples of physiological processes can correlate to define an evolutionary response. It was the case when somatic maintenance and energy conductance correlate because it improves the fitness despite reduced longevity. Moreover, if we talk about the environment, temperature is one of the known environmental stresses that can increase the vulnerability of organisms by reducing their resilience and resistance. And we saw that increasing temperature leads to higher intrinsic rate of increased air prime and a shorter lifespan, which match with, as we can see in literature. So this assumes that temperature drives fast strategy organism with rapid growth, early puberty, and shorter lifespan. But some limits occurs in this model, and the first one is the stimulating choices, because at the beginning we vary the parameters one at a time, or we did a combination of only two parameters. So a better representation of the evolutionary potential can be obtained by varying all the parameters simultaneously, but this is a long simulation time, about 8 months. Another limit is the behavioral issues which can occur, like for example, in depth theory, somatic maintenance considers the energy expended in the movement and displacement behavior. So here in the diagram. So a spatial study might be relevant to see if individuals are able to change their habitat in response to climate change. And this is particularly relevant for species such as salmon, because they have a migratory behavior. Because, okay, so here, this is the data used for the primary parameter estimation for salmotruta. And this is the reference used. And as you can see here, there is some references about freshwater trout. So a fish will spend its whole life in freshwater. And in blue here, it's reference about the sea trout. So salmotruta, which migrates to the sea and the ocean. So, but these two individuals, even if they are in the same species, they have difference in habitat and food types. So I re-estimated the parameters with some reference just about sea trout or freshwater trout. And I saw that here there is huge difference in energy conductance and somatic maintenance. So all the energy in this part of the diagram. And moreover, here there is huge differences in life cycle related parameters. So for example, the different maturity threshold and also the shape coefficient, which prove that there is a difference in habitat and food types, and that we need to re-estimate the parameters for the population we want to study. So in conclusion, temperature influences fish physiology, for example, the metabolism, stress response, or also life history traits. We saw that fish exhibit adaptive strategy thanks to the physiological processes, fish and, yes, freshwater organisms in general, I study. And also that the model is useful to predict the evolutionary potential of organisms. And moreover, further research is needed to understand the genetic implication. Like, for example, in throat, there is a gene which is responsible to the age of puberty. So it could be really interesting to link the real gene to the biogenetic processes, like the primary parameters in the depth theory. And moreover, we need to implement some performance interval response, because maybe sometimes when the temperature is really high, the organism will not have the same thermal response comfort. And finally, to consider the behavior is needed to, as I told you, with the sea and the freshwater trout. So now it's the analysis part. So I would like to thank firstly the thesis jury to accept to evaluate my thesis. I also want to thank the thesis committee, which followed my work during these 3 years. I want to thank like the no-pass committees, because I organized these seminars and it was like a really great adventure. I also want to thank the DEB school, where I learned a lot during almost 2 weeks. I will switch in French right now, but je veux vraiment remercier Mélanie de Saint-Pé-sur-Nivelle en général, mais particulièrement l'équipe EcoBiop, avec qui j'ai toujours une super ambiance et avec qui je me suis vraiment sentie très très bien. Et je voudrais remercier particulièrement Charlotte, qui m'a encadrée pendant 3 ans. On a toujours eu des discussions genre super stimulantes. Je sortais de réunion, j'avais encore plus d'idées qu'avant, et enfin vraiment ça a été une super expérience. J'aimerais remercier Valérie qui a accepté d'être dans mes supervisions tout de même. J'aimerais remercier mes 2 stagiaires qui m'ont beaucoup aidé, notamment pour la réalisation d'un des chapitres de cette thèse. Bien sûr, je veux remercier tous les doctorants. Je n'ai pas trouvé de photos où on est tous, mais particulièrement le Oshashunagang. Et j'aimerais remercier notre mascotte Ernest, quand même, qui m'a donné beaucoup de câlins, surtout pendant l'écriture de mon manuscrit. Voilà, so thank you all for your attention, and I'm open for questions.
Une personne veut sortir avant les questions.
Okay, so now we'll start with the questions from the first reporter. So this is Dina.
Hello.
No, we can't hear yet, but let's see. Oh, now we can. Okay.
Okay. First of all, I would like to thank Mélanie for this clear presentation. It was really very well done. The topic of your thesis is both interesting and original, and bridging the bioenergetics and evolutionary strategies is a very complex task. I think you have made really a valuable foundation for the future developments. A few words about your thesis. Your thesis is well written and well structured. However, there are a few technical details that need to be addressed, and I think I put most of them in my report with comments and suggestions. And now I have a few questions to explore some of your ideas you presented today. First, you have started to investigate how variation in 6 of the primary DEB parameters influence some key traits. Could you elaborate on how you decided to select only these 6 parameters? For instance, why you excluded parameters, the life cycle parameters, as you mentioned at the end, that differ between species, and also the aging parameters. Do you consider these parameters to be less influenced by evolutionary pressures?
No, okay. At the beginning of the thesis, I wanted to focus on these parameters because it was the parameters the more present in the energetic fluxes. And so I wanted to focus on these ones. But yeah, I remember that at the DEB school, we spoke about this, and I thought it was a good idea to take into account other parameters like related to the life cycle. But as I started the PhD, the thesis with this 6th one, I wanted to stay focused on this 6th one and yeah, do more results with this one. But actually, if I have time, I would like to explore the other life cycle related parameters, and especially the age at maturity, because as I told, like it's known that gene can have an influence on the age at maturity, for example, and it would be like really interesting to link this parameter like EHP, you know, with some real gene in the salmon, but so yeah, I would like to, but
okay. Okay, I understand. Yeah, so yeah. Also, you mentioned that some of the parameters, you said that you vary them maybe widely. For example, like the volume-specific cost of structure. The range that you varied is from almost 1,000 to 25,000 joules per cubic centimeter. And you know, of course, that this parameter is linked with the growth efficiency. And typically this growth efficiency is around 80%. So if you have this large value for this parameter, the EG parameter, then you would get as a result a very, very low growth efficiency. How you would explain this in your analysis? Why? I mean, you had to vary it that
I'm
long.
not sure that I understand. Like, you mean why I choose to vary them, like in the range of possibilities, even if it can cause some disruptance with the expression of traits?
Yeah, I mean, you take a value of this volume-specific cost of structure up to the 25,000. And I think this is a really very large value. And that would cause the growth efficiency to be very low. Less than 1% or something like that.
Yes.
So, have you considered that?
Okay, so I chose a range of possibilities because I was not expecting some natural values or natural things that we can see in nature, because it was really an exploration about how the values of these parameters can impact the expression of traits. So yes, even if there is some confrontation, like even if it's not natural, I thought it was maybe quite interesting because it can bring a clue about the evolutionary potential, even if it's not really real.
Yeah, because I mean, this question relates also to another question I have. For example, when you look at this other measure of fitness, like the intrinsic growth rate, when you vary that parameter, the EG parameter, widely, then you see some discontinuities in this maximum values you compute. I don't remember if you showed it in your presentation today, but it's figure 4.3 in your thesis.
Which one of the heat maps? Those when I cover with 2 parameters or just one?
No, it's just one parameter.
Here,
Okay, I want to.
you have it here if you want.
Yes, but I have it on my computer, but I don't know how to change the slide because it's.
If you fais Alt,
Okay.
you can do that.
No, it's in the other.
It's accessible.
Yeah.
So with okay, so the energetic rate of increase here, and for energetic cost of structures, this one in the middle.
I don't see it now. I think
sorry.
I lost.
Okay.
I think she's mentioning the discontinuity in the triangles.
Yeah, yes. Yeah, in the triangles, yes.
Okay, so you think that as the value is like really high, it could cause this discontinuity, right? Because it's confronted with the estimation of growth.
Yeah, so I was trying to understand why you get this discontinuities in some of the parameters, the variation of the parameters, like and that happened only in the intrinsic growth rate, not in the other.
I think this is
traits.
Because if the energetic cost is like really high, yes. But if the energetic cost is really high at high temperature, like we saw that high temperature, like the best fitness, the best strategy is to be fast. And actually the high energetic cost for structure can be related to a fast strategy because the individual will have a high metabolism and will expend a lot of energy in structure. So yeah, for me, the best fitness will be at high cost of structure because it means that a lot of energy is required for that, is mobilized for that.
Yeah, I'm wondering whether some numerical issues happen there because for all the other graphs, you see some continuity in these triangles, while for the intrinsic, for the population growth rate, you see a lot of discontinuity somewhere. But yeah, okay. Yeah, also in these graphs that you are showing now, you mentioned in the legend that you normalize them with some maximum, I guess. And
okay, it was for Salmotrota?
For both, I think.
For both?
Yeah, even for
yes, even for Daphne, sorry. Yeah. Yes, it's because in the other, when I didn't normalize, I didn't see any variation, like there is no color on the heat maps. So yeah, I choose to normalize and to do a rate with the intrinsic rate of increase.
So the
best value.
Yes, sorry.
Sorry.
Yeah. So you divide it by some maximum value, right? How did you produce that value?
Actually, I did the rate, so like the best value is 100%, and the not best value is 0%, and so yeah.
Can you repeat that? I didn't.
But I did a rate with the intrinsic rate of increase. Like for example, the best value is 100%.
Yeah.
So and the lower value is 0%. And so yeah, it was the only way to see the influence. If not, we see the triangle, but not the colors.
So you divide by the maximum, taking it all over the simulations, not only for each when you do one parameter at a time. That's what I understood from the graphs.
Yeah.
Because otherwise you would see this purple in all graphs.
Yes, I did for all the simulation at the same time. Yeah, exactly.
Okay.
Not individually for each graphics. Yeah.
Maybe you want to say that in your
in the manuscript.
Yes, in your manuscript.
Okay. Yeah. It's a good one. Thank you.
Yeah. And also, my last question is regarding the way you calculated all this measure of fitness, the lifetime reproductive success, the intrinsic growth rate. The lifetime reproductive success is calculated using the reproduction rate and survival.
Yes.
Those quantities are continuous, but you presented them as discrete, as a summation of the product of the two.
Mm-hmm.
What type of approximations you have done to do that? And yeah, also for the growth rate, which is usually calculated via the Euler-Lotka equation.
In my simulation, I use that as a discrete because like the calculation of the reproduction output was at the end and the survival was also at the end. And so this is really a calculation after simulations. So yes. I don't know what you mean after that.
Okay, it was not just clear how you did it this approximation. Okay.
Okay. Okay, I will make it more clearly.
Yeah. And my last question is that if you were to start your PhD now, with all the experience and all the insights you have gained from this work, all this, I guess, 3 years, what you would do differently? Are there any specific aspects of your approach that you would change?
I think I will. I was maybe a little bit naive at the beginning of my PhD because the depth theory was really new for me. So I took these parameters and saw all the influence, but maybe I will focus just on less parameters regarding this one and include other parameters like the life cycle related parameters, for example.
Mm-hmm.
And so maybe I will change the selection of these parameters and going straight to the fitness, or maybe less explore some morphological traits than I did. But at the same time, it was really useful for me to understand all the mechanism of the dev model. So it depends if I know the dev model.
I wouldn't have skipped that one because you really gain insight when you do this exercise. Okay, thank you very much, Melanie.
Thank you very much.
The questions.
Okay, thank you, Dina. So I think I will follow up as a second referee. So I also want to thank you for inviting me to review your PhD. It was super interesting, quite a challenge for me, I have to say, because I like phenotypic evolution, but I usually like very black box models that don't go into mechanisms a lot. So this made me think about how you can include these mechanisms. And I had lots of curiosity, also lots of challenge trying to understand. And I see that people practicing it also have these challenges of interpreting all these things because there's so much going on in the model. So, I mean, I want to start by very general questions like, for instance, even some basic parameters were not that easy to put in words. Like, for instance, this energy conductance V dot, you know, broad meanings, I sort of see how it is, but have people really, when you measure it, how does it mean biologically and how does it relate to this energy conductance parameter?
The energy conductance is all the energy which is like in the reserve will go into all the physiological processes because it can impact the growth and also the reproduction and also the maintenance.
But biologically, in terms of something you measure or?
For me, it's like if we take all the fat to build muscles, for example, so it can be linked to the movements and behavior and yes.
Okay. No, I was just wondering how it's measured.
Oh, it's measured.
Yeah, this type of things.
It's
for the structure, I can see what it is, the cost of structure, but things like that, it seems like almost. But seen from outside, it seems almost philosophical.
For example,
energy flow, you know.
If I'm not wrong with the respiration rate or something like that, this with metabolism measure.
Okay, and I don't know the answer. I was just asking. Also, something that strikes me a little bit about this theory is that, for instance, the equation for senescence is all about accumulation of damage, and it seems like you can escape senescence by growing and diluting your damage. This is part of the model, right? You're discussing this at several times. So is there evidence for that, or is this really supported, this idea that you can keep growing and then dilute your damage and not grow old, basically, diluting the damage?
But it goes like a lifespan which is like later if we don't put energy in the somatic maintenance. Yes. If there is an evidence about.
Yeah, I'm just wondering what supports these types of assumptions. The big assumption that
basically the flow strategy. If the individual will not put a lot of energy in maintenance, it will have a later lifespan, later age death. Sorry.
But this idea that you can grow and that this will dilute the damage and cause later senescence, is that what you're describing now?
No, I spoke about if there is less reparation of cellular damage, it's because there is less energy in somatic maintenance, and thus the energy will grow more, and the lifespan will be later, but the age of death, will be later, and it's like the flow strategy in organism.
Okay. Then in terms of the responses that you get in your first chapter, I was wondering, so it's kind of the same types of questions, always how you can try to make sense of these things that depend on so many parameters. Like, for instance, for age at puberty, it differs completely between the response of varying PAM, the assimilation rate, and kappa.
Yes.
They're all the same, except that the age of puberty is completely on the opposite direction. So can you explain that? In your chapter 3, the first, when you study you vary the parameters individually.
Like here?
Yeah, like here in the bottom, there's this blue and orange for age at puberty.
Yes.
There's kind of the main difference, and all the same. The rest is doing the same. So you came with an explanation. Perhaps you can remind us the explanation for this.
Yes, actually, if the assimilation is really high, so a lot of energy will be in the reserve, but the kappa is still the same. The fraction of energy will go on the structure. So the individual will have will be really tall and will have a high weight, but the other fraction kappa, 1 minus kappa, go on reproduction, but so the age of puberty is earlier, because there is like a lot of energy will go through all the diagram, and so the maturity threshold will be reached like more early than for the other.
And so in the other case, when you vary kappa, it's the reverse.
When
I'm comparing these two effects.
Yes, because a lot of energy will go through the structure part of the diagram. So less energy will go on the maturity and reproduction, and thus the individual will reach the EHP threshold later. Yeah.
So it's for lots of this explanation, I was wondering whether, I'm trusting you on your interpretation, but there's lots of this interpretation. Is there a way to show what you're saying? You're invoking some steps. It's affecting that and then that and that. Is there a way to show this path that you're describing, you think? This causality path. You're describing a causality path. You're saying, I'm varying this. It does that because I think it changed this, that, and then it led to that. There's lots of that in your PhD. I think I'm not saying it's you. I think it's in these types of models. I'm wondering if there's a way that you can show other than verbally, because you cannot do I don't know if you can do the analysis mathematically. It's too complicated.
Yes.
I mean, I'm used to simpler models where you can have an analytical solution and say, is this that's causing that? Here, you can't, I guess. It's so complicated.
Yeah.
But you see what I'm trying to get? Is there a way to show?
Is way to show, but
this intermediate steps?
Or
anyhow, I don't know. Some path analysis, you know about path analysis, right?
With the diagram, because I see like the movement of energy, but I'm not sure that just with the equation.
How did you do when you were interpreting all these interpretations you gave? How did you reach these interpretations? You use it's a real question. I don't know.
But for me, it's kind of logical when you get like, I don't know how to respond to this, because for me, if more energy will go on maturity, of course, like it will have earlier maturity.
But there are some complex things like this yellow one in the bottom. Is like if you allocate too much to reproduction, then you don't grow enough, and then you don't acquire enough resources, and then you don't, it's a complex.
It
involves, it's not a straightforward line. There's feedbacks all over.
Exactly, there are some feedbacks because as the individual, for example, can reproduce a lot, but the individual is like really tiny, like he cannot produce 1,000 eggs if he's just few millimeters, you understand? So yes, of course, there is some feedbacks between the structure and the reproduction capacity or maturity.
So that was kind of my question. How do you keep track of all these things that could affect your traits? I mean, is there a method or you just...
It's complicated. I don't know, I just try to think about it, but to track it. Like actually, I did a lot of this kind of graph, and we tried to understand why sometimes the curve is with this trend or this trend. And sometimes I just add like all the mathematical equations of something and try to link things. But
can you shut down, like for instance, here you just change one by one. And when you change one, can you also shut some path and say, I'm trying to just this one act and see if these types of things, reduction is...
I think we can like...
Or is it
record all the steps of the model, but I just take the primary parameters and I quantify the traits, and I did not output like the interaction.
But then we have to trust you on your interpretation. Sometimes I was willing to trust you. Sometimes I didn't know.
Yes.
I don't know. It's just comments.
Actually,
anyway, so in your okay, I'm going to skip in the next chapter. So it came more to a link with ecology, with this fitness proxies. So I was wondering, what's the difference between these fitness proxies, or which one will matter for evolution? In which you have LRS versus intrinsic rate of increase. I mean, which one matters for the organisms, they don't know if this is my LRS, this is what they feel is like something will affect their evolution or not. So when will each of these you think matter more for evolution?
Between the LRS and the R prime. Okay, I think the LRS is more related to the reproduction, so it's kind of important, but the intrinsic rate of increase will be more related to all the life and of the ability to the organism to transmit the gene and maybe, no, not maybe, and especially the intrinsic rate of increase can be used for population model, something like more precise. So I think the intrinsic rate of increase is a good explanation as it uses the LRS, but we can determine some evolutionary profiles with the LRS.
It also depends on the ecology, like in expanding population, basically.
Yeah.
The R is more relevant because if you reproduce faster, you outcompete the others. When if you're a more stable population, then there's not much point in doing faster. You have these issues also that are important ecologically. For the effect of temperature, so lots of the effects seem to come from this Arrhenius type of equation. And you assume that it was the same for like 3-4 of these parameters that basically have the same equation. I have the feeling that that's what's driving, for instance, the R prime, which is also something about speed, you know. You increase the speed of lots of these processes, metabolic, and then it increases the speed of growth. So, but I don't know if we have, what's the rationale for using this equation for this? Why did you choose the traits that are going to affect that?
By the
Temperature,
temperature?
yeah.
The Arrhenius.
Yeah, how did you choose, we're going to apply this equation to this and this trait?
But the Arrhenius temperature was already estimated like in the database, so I just used the estimated parameter. So
we're confident that these traits, these parameters are going to vary in that way with temperature.
But it's like a correction to the actual.
Yeah, I understand, but there's a correction, but then there's this equation that has an exponential of temperature in it.
Yes.
That's what I see from it.
Yes.
And so you apply it to 4 of these parameters, right?
Yes.
So why was it this 4?
Okay, it was set like that in the depth theory.
Okay.
And I have to assume that I didn't.
Yeah.
Not I look at it.
Yeah.
So I just trust in the theory.
That it's basically some rate of activity that's going to be increased with temperature.
Yes, yeah.
So yeah, this was interesting, but again, usually with people who are not specialists, they ask the most nasty questions. I know a little bit about that, but when I look at tolerance curves, usually I see them going down, and there's CT max and things. It's a big issue. So here, it wasn't in your model, which is you assume you're on the left of that, okay? But since it's these models are really mechanistic and things, I was wondering if you had any idea of how you would bring this kind of breakdown at high temperature in these models, where they could act, you think?
Breakdown at high temperature, but at which point?
You know, this idea that you see, fitness in general as a function of temperature increases and then it collapses usually.
Yeah.
And this was not in your model, right?
Yes.
How could this come into your model, do you think?
Because I don't have any thermal tolerance.
Yeah, but how would you bring that in the model in terms of mechanism? It would be, I don't know, accumulation of damage or if you were to do it.
I think I need to implement some reaction norm to temperature, but for that I need to study the response norm of the individual, the species I want to study, and it's something I wanted to explore more during this thesis, but I didn't have
Yeah, sure, it's difficult.
time required. But
in terms of all these processes, which one do you think are be more likely to be affected if?
I think the maintenance, because if temperature really increase, the maintenance will be really increased too. So because this is the process which lead to a reduced lifespan. So
okay,
yeah, for me, this one, and yeah, an acceleration of metabolism in general, like, so maybe energy conductance too.
but then it has to be costly.
Yeah.
So is it this cost of higher metabolism, you think, would be the reason?
Yes.
That lifespan decrease.
Yes.
Okay. Which time? I had also this question about discontinuity that struck me on this optima, but I'm not sure if it's a numerical issue or it's just that the optima, it might be that it becomes very flat, your fitness landscape, and then this thing. I was surprised that it's only for R prime, for R that you see that, not for LRS.
Yes.
And also another related question was that in some cases, the optimum is right at the boundary of your parameter values. So I was wondering what does this say? Does this say that you picked up some limited range of values or something? The model wants you to go more to...
I don't think the range of values is limited because like there is some values which is like really, really, really high or really, really low compared to those estimated in the database. And yeah, there is some values which is not like naturally observable.
Yeah.
So I don't think it's a limit in the range of variation.
Yes, true. And you argued for that because at some point in your one of your last chapters, you were saying we excluded some life histories because it was so fast that we could not estimate the metabolic rate, the scaling exponent. So then this made me wonder, going back, how many of your life histories were realistic, Yeah. So I understand your point that you want to explore the potentiality, sure, but did you consider also sort of restricting to more realistic or values that are consistent with observations? Did you consider doing that too, or?
Yeah, I considered that, but at the beginning of the thesis, I didn't know how to, like, for example, in this range, in which range of assimilation it's natural, because it's not really an observable trait, know. So I wanted to do the range of possibilities just to see like evolutionary potential. But now that I have a better understanding of how it works and the mechanism, maybe it could be like really relevant to see like at which range it is like natural, like observable. And I think for that, to compare between different species should be relevant. And for different species with different ecology or something like that, to restrict the range of variation, of course. But for the three chapters of my thesis, not the last one or the rest, it was really specific to a species, so I didn't compare to another species. So it was like the range of possibilities. But yeah, it can be relevant.
Okay. On the chapters with the pairs of traits, I had this question that I asked in my report, but I guess you've sort of touched to that in your presentation about interactions being more than the sums of effects. So I guess you've changed that. You've changed the way you're thinking about this. Because this made me wonder at some point, it seemed like the effects were additive, and in the sense, it's important to know that they can both act, but if they have the sum of their effects, then it's not the same as this.
But actually, we we had some discussions about this, and yeah, I think like for that, for example, it's of course it's quantitative because we have the value of the parameters and the value of the intrinsic rate of increase, but I think more explanation and more simulations are needed to really see, like, if example, like to see if there is additive, like we take a value and see if it's really the additive effect, because here we can just see if there is a change in fitness landscape or not, but it brings new idea about this, yes.
Yeah, if it's additive, even here you would see some straight lines basically, which you see on the bottom, for instance. When it's like that, it makes it looks like something additive, perhaps.
But we don't know if it's really additive. For that, we need to see to know the exact value of the value of the
for each of them.
Of this value and the value of the fitness of this value, and see if it's additive or just an interaction or just the same value of fitness or something.
Okay.
So, but yeah, actually, I really want to explore that. It's quite of interesting.
So something else about these correlations, or this is like more, what would be called correlational selection, how fitness might favor combination of trait values, right? So it's interesting, and also the correlation among traits or secondary traits, they emerge from what you put in the primary traits involved. So, but which is satisfying, but then you can go. So one of the problems is that you push the problem one step farther. You would say, but what are the correlations among these primary traits? Can we know something about this? Because this would tell you how they would be selected. If some combination of primary traits lead to higher fitness, they might be selected to become correlated. But can we tell if they are correlated?
Correlation between traits, right?
Between the primary traits.
The primary parameters?
The primary parameters.
Oh, okay.
These things, when there's a combination of these primary parameters that had higher fitness, this would select for them to become genetically correlated, for instance. But I don't know if it's easy to show empirically if they are correlated or not.
But yeah, but I think we can do that with the quantitative study that I told earlier. But I think it's really possible. And yeah, it's something like which can be like really interesting to explore. But yes, it's completely possible. And I was expecting, for example, for the different colors for the additional effect, and sometimes like no colors, like when there is no effect on it because the values are the same. Like, but yeah, I think it's really possible. We have kind of long simulation time, but yeah.
But I was also meaning empirically, without measuring fitness. So can we measure the correlation between kappa and PAM, for instance? Can these things be measured or estimated and across species?
Yes. Yes, it's possible, but it depends of the ecology. I think like the correlation between two parameters depends too much of the ecology of the individual, and so maybe for the same individuals with species with the same ecology, it can be interesting, like to do a correlation, and after that have a value of the correlation, and maybe it's something that can appear across species, but I'm not sure. It's
okay. Perhaps you didn't show your last chapter. Can I ask you questions about that? The one on allometric relationship, because this is like
if you want.
even I had heard about this. So, and I know some of the authors of this white paper that you're citing. So I was trying to understand because the authors of this, it's this paper that's less mechanistic in a way, the white and tag paper. They just assume some different allocation to reproduction at different ages.
Yeah.
And they somehow imply that having a fixed kappa in your type of models constrains the allocation too. Is it the case? I'm wondering, I'm not sure. How does their F parameter, that determines how much you allocate reproduction versus
The F?
F, it's called, right? The parameter that determines in their model how much you allocate to reproduction.
In the paper of White?
White, yeah.
I don't remember the name.
Yeah, but you know what I'm talking about, I'm sure, right?
Yeah.
Yeah.
A little bit.
Yeah, they said that you allocate more or less to reproduction as a juvenile and then as an adult at different ages, something like that. In the DEB type of model, when you have a fixed kappa, can you still allocate different parts of your energy to reproduction at different ages?
Allocate in terms of fraction of energy, because kappa is like a fraction of energy. So if the kappa is fixed, like it's the same fraction of energy will go on.
Yes, that's what it seems, but I have the impression that with all these feedbacks, with growth, etc., there can be more going on. I don't know. I found it not that easy to relate the fraction of energy that you allocate to reproduction in a static model, static in the sense that it's assumed, versus dynamic model like that, where there's all these feedbacks. This is a question that I had reading this chapter, but I don't know if you have a...
I'm not sure I understand the question there.
Okay.
Like, sorry.
It's not precise, I guess, perhaps not precise, but you're trying to confront your model that has this dynamic thing with their model, which has a fixed, if I understand well, proportion of energy that's allocated to. But I was just wondering if the amount of energy that goes really into reproduction can change, even if you fix the kappa, can change among ages just because of all these things that happen in these models that are complicated.
The quantity of energy, for example, in reproduction can change if, for example, there is a lot of dissipation process. But for me, if the kappa is fixed, and the quantity of energy going on the processes can change, but the fraction of the energy, the mobilized energy, is fixed.
Yeah. Okay. And in this chapter, you found that the scaling exponent that had the highest fitness was quite low, actually. It was much lower than what people found empirically, isn't it?
Yes.
So you found things like were the ones you had where you could show the relationship with fitness. I don't know if you have this curve.
Which one?
The one with the fitness, this one.
Yes.
Yeah, the maximum, well, we can see it on the right, I guess. The maximum was for small exponent, right? Or on the top there. It was for an exponent of 0.3 or something, so.
Excuse me, which one exponent?
The one that's here, the yellow thing, it's near an exponent of 0.4 or something, which is lower than what people find empirically, right? So how do you explain that? I think it's...
I explained that because in the juvenile stages, they don't have the same use of energy for metabolism and growth, because, like, for example, for juveniles, like, they need to grow for avoid maybe predation or something. And so when the adult stage is reached, there is like a stabilization of this, of the allometric relation, and thus the exponent is lower.
Okay. It's also the fact that you use this kind of ontogenic allometry throughout life, perhaps not exactly what everyone measures, because this was an important component. It's age-specific. Yeah, I think it's normal. Okay, I think I'm going to move on to the next person. I also had some kind of general question. I mean, I'll just end up with a general question, a bit like Dina. Like, Or it would be like, what's next from there? Is it going more complex or trying to find some ways to simplify it in this type of...
If I have to continue my...
Yeah, if you want to go on, do you make it more complex? Or do you want to try to simplify it, or even within this model, try to find ways to, I don't know.
I think I will try to simplify a little bit, because yeah, sometimes I was kind of lost with all the parameters and all the traits and all the trend, all the relation, and maybe to focus on some parameters of interest. Maybe it was not so useful to take all this trend, even if it leads to an exploration, which is interesting. But for more research, for further research, I think I will more focus.
Yeah, also related is, if you want to apply this, these were like supermodel organisms like Daphnia, etc. If you want to tell people, okay, you're going to apply this approach to your model organism, which is not, how do you translate this to less well-studied organisms?
To less studied organisms?
Yeah, not everyone works with Daphnia.
I think I will just re-estimated the parameters by my own, like with some data on a specific species or something like this. So yeah, I think the re-estimation of the parameter could be interesting for when we want to study like a specific population or something.
Can't you identify like the big parameters are likely to work and say, oh, perhaps you should focus more on this and that?
Yes, maybe, like, yes. Actually, I compared my work with some people who had the same problematic as me, but in an experimental way. And I was pretty happy that I had the same results as some people who worked on Daphnia, for example, and had the same results as me that I did theoretically. And so I had a great discussion about that during seminars. So.
Okay, thank you. I think I took too long, so I'm going to leave questions to Gonzalo now.
Okay, thank you, Melanie. It was nice reading your thesis, and it was a nice presentation. So my first question will pick up on a question here from my colleague. We was asking, how would you measure the energy conductance? Yeah. And the question is, can you measure the energy conductance? Do you think of a trait that it gives you the energy conductance in some way?
No, I don't think there is like just one trait which can be used to estimate the energy conductance, but it can help to have an estimation, at least for a range for the energy conductance, if I'm not wrong.
Okay. So in general, how do we get numbers for these parameters?
How do we get what?
How do we get values for these parameters in the depth theory?
Yeah, it's from the zero variant data, and after the estimation, I'm kind of trying to remember exactly, but.
So you do an estimation procedure.
Yeah.
With all the parameters involved, right? You don't estimate one parameter at a time.
Yeah.
Yeah. Yeah. And why do you think that is? Why don't you estimate just energy conductance or just the PAM?
Yes, so now but we have to estimate all together. It's not really possible to do it independently.
Yeah. So you don't have traits the traits have different information from different parameters, but you don't have one that gives you information good enough for one parameter or another. So it's not possible to have an idea of any of the parameters just by one trait. It's more complex. Yeah. Okay, can I ask you to go to slide 42? Is it 42 or is it? I'm looking for the slide where you have the impact on R prime and
With
yes,
yeah,
that one.
yes, okay.
Yes, that one. Okay. So when I look at this on the top left, R prime and LRS there, it seems to me that there are two behaviors there where you have a very well-behaved phase, right? And then you have then a misbehaving phase, let's say. So you have dots that are just in a line,
okay.
and then you have some scatter, okay?
Mm.
Seems like there is kind of a phase transition from one to the other. What do you think is happening here in this change?
Okay. I think here, like the temperature is really high for the individual, and here, like maybe the amplitude is not so much high for the throat because it's like 10 degrees, but 10 degrees around the mean. So it's just like a seasonality event or something. So here we can see like a disruption because just of like really high and hot temperature for this organism.
Yeah, but connecting with what was said, so you have this temperature correction that is an exponential temperature correction, right? And when you do this temperature correction, there is no disruption there. You don't have high temperature problems in the correction that you use. So where could this disruption come from?
Because of the estimation of our reduced temperature, which has to be like, no?
I don't know, because you didn't estimate it, right? You used it.
Yeah.
Just a value. Yeah. I'm not sure. I don't have an answer for that. For me, this is interesting. This is a result that is interesting, and I would like to understand better what is happening. But let's.
Actually, there is more variability also because there is like more individuals than here. As you can see here, there is like 100.
Yeah.
So
but still,
maybe if like I have like maybe if I just increase the amplitude here and continue the simulation, we will see the same trend.
The problem stands because you have to then answer the question on the other question. But yeah, so I would submit that this is an interesting result that you have to think about what's happening here. So, but let's talk about the first phase where the things are ordered there. And you see the effect of the temperature, and you have this result, and you come to the conclusion. You say at some point, say hotter is better. So because the individuals get quicker, but is this enough in the sense? So think. So all the rates are increasing, right? Because of this temperature increasing. All of this is increasing. But now suppose that you have the species of Daphnia and you have another species of Daphnia. They have also temperature increase, their rates also increase. So is this Daphnia better positioned compared with another Daphnia species?
I do not understand the difference between the two Daphnias.
Let's suppose they have different parameters.
Different parameters?
So yeah, so different species, so they have different parameters.
Okay.
And you have this Daphnia that is getting things are getting faster, the reproduction is getting faster, and the other Daphnia, if you do the same type of thing, you will also have it being better. So when you say it's better, this is better for this acne, but is it better is an absolute, or how do you say it is better?
Okay, in this case, it's better for like, here it's only individual with the admired pet parameter value, you know. But actually, like, author is better for fast individuals. So if there is any individuals with values of parameter which is more related to a slow strategy, maybe it's not better at all.
But now, suppose if you have two faster individuals.
Too fast.
Two
Okay.
fast individuals, and now you say it's better. But if they are both more productive, how do you say that it's better?
There is no one better than the other. They are just like, they could fit with the given environment, which is like with a temperature or something.
Mm-hmm. So do you think by like looking at this graph that you could say that it's better, this organism, this
so.
species? Yeah,
I think so. Yes.
So if suppose so my idea is this: you have something, you have a film of two species reproducing through certain time, okay? And so if the only thing that the temperature does is increase, what you do is that you put this film in high velocity now. Okay, it's the only thing that temperature does is increases the velocity of the things that are happening. Okay, so if the slow film or the film is now running fast, is it better for? Can you say that it's better for one case or the other in terms of comparing the species? If
comparing species.
Comparing species.
You speak about something like I'm
let me see if I can do this in another way. What does it mean to be better? What does it mean to be better?
In this case, it's like when the hair prime is like higher.
Okay, so it's reproducing more. Okay, but if why is reproducing more better?
Because maybe it has like early puberty and can reproduce.
But suppose that one species reproduces more, okay, and another species also reproduces more. In this situation where they both reproduce more, is this species better now?
No, okay, yes. But yes, it reproduces in other cases. Like, I don't get it.
Yeah, but if it is comparable, could it be that they are just in the same situation? Because everyone is reproducing more. Suppose everyone is reproducing more, so it's.
But the genes is still released in the environment, so.
Okay, so it's a philosophical discussion on what is the meaning of the R prime and the LRS. So think about it if it makes sense in some way. Okay, let me change to another idea. If you go to slide 15 now. It's the slide where you put the models. Yes, here. Okay. Here, can you explain me the green box here?
Okay, so it's because like the energy cost for structure has an influence also, yes, of course, on the structure, but also on the mobilization energy. So the box is just to include like the structure and the mobilization.
Okay, so it doesn't include the somatic maintenance?
No.
Okay. I see.
But yeah, actually the somatic maintenance depends on structure, and structure depends on energy cost. So it was, but yeah, no, for me it was just to try to highlight these two fluxes. It's for that I detailed the calculation with the list on the left.
Okay, but for example, the assimilation also depends on the structure, right?
Mm.
Yeah, and you didn't put the box of the assimilation.
Assimilation depends on the structure.
It does.
Yeah, it
right?
does. Yeah, it's true. No, actually, I just thought about like the mobilization flow and the structure, but yeah, maybe the box was a little bit misplaced.
Very well. And if we go now to slide 29, let me see if I. Okay. I like very much this scheme that you try to look at the effects. Yeah.
So you need to navigate.
Wait, I'm waiting now. No, 29, yes. Yeah.
We cannot see the slides. Can you share screens?
Why should be like that? Yes.
Yep. Okay, so the idea here is you colored the lines that were affected positively, right? Increase, and then you use the dots to say they decreased, right?
Yes.
That's the idea. Okay, so looking at the first slide, what captures my eyes is that you have one line that is not colored. Why is that?
This is Yes. It's because I was not really interested in this part of the graph, and I assumed that
Okay, you are not interested. Okay, so now you would color it.
I think so.
Mm-hmm.
Yeah.
Okay. Very well.
Yeah.
Good. And if when you do, for example, here in the somatic maintenance, you increase the what goes into the somatic maintenance, and you say you decrease the what goes for growth, right? And then when we look at this from a life cycle perspective, what is the impact of this decrease in structure?
In life cycle.
The fact that it decreases, so what is going to happen in the life cycle of the organism? Then it will affect other flexes or not?
Actually, it will affect the reserve. So of course, if the reserve cannot have like a lot of energy, the energy which will go into the reproduction and the maturity, yes, it can affect the life cycle. And if we have less structure, we will, the individual will have like later puberty. So yes.
Yeah. So you are putting here the direct effects, right? And then there are indirect effects like the one that you were also discussing here about how do you make the causality, how do you go through the causality of what is happening here. And when you, so for example, in the case of the kappa, where you have colored, look at the colored lines there. Do you think all lines that are colored should be colored?
No. Like the line here. No, yes, the line here has been colored.
Which one?
I hesitate right now. Is it like, okay, the somatic maintenance should be not colored? Because it's just like the structural V and the PA.
Yeah.
So, and there is no interaction of kappa on it.
It could, yeah. That's the direct effect. Yeah, it could not be. But that's what I really like about this scheme. It's that you are trying to show us what are the effects, but you have to really be careful about the things that you choose here.
I will modify what.
But continuing on this graph, so what happens is what you're describing here in terms of the decrease, the part that is increasing and decreasing. But if you have an indirect effect because of the structure, so if the structure increases, can you say something about what's happening in the other side of the?
Less energy will go toward the reproduction.
With the increase of the structure.
With the increase of the structure, but it's the same way. Like the reserve can be higher, so higher energy here, but the fraction of energy will be like in majority go to structure. So yes, maybe the reproduction can be not that bad. Yeah. But yeah, still increasing a little because of the increase of reserve. But here I wanted to highlight that more energy is still. Yeah,
so you are highlighting the direct effect of the change. That's what you're trying to highlight, right? The direct effect. But maybe it can even increase the reproduction buffer, depending on the parameters, depending on how much you change. Maybe it can even increase. So it will be nice if you be very clear what you are showing with this scheme, because I think it's a really good idea. And talking a little bit more about the indirect effects would also help in showing what is happening here. Okay. So now. I'm not sure if I wrote. Did I talk about slide 47? Because I don't have here.
47?
Yes.
This one? Yes, this one.
Yeah. Let me ask something here. On this, it's also connected with was already talked about the discontinuities that sometimes appear. And here you have a band, a yellow band on the frontier also. Do you have an idea what is happening here on this type of graph?
For me, if the assimilation is very low, like the energy conductance like is still high, there is no enough of energy going to the structure and the reserve, so the individual is like limited by its quantity of energy in reserve.
But why do you think this happens so quickly? So if you go a little bit away, so you see you have this yellow band and then you have bright red. Do you have an idea why this is?
For me, like it just make an evidence of like the limit of the individual to be able to conduct energy to the structure and the reproduction, and thus like it's just not possible with this value of assimilation to just make the structure.
Mhm. Okay. Yeah. Okay. Now just a more general question about in the introduction. At some point, you say this. So it's just let me have it here. Incoming energy is stored in the reserves in the form of lipids. Okay, so when we are talking in depth theory, is the reserves always in form of lipids? But
Sorry,
the reserves are always in form of lipids.
is it a question or a question?
No. You say that here it's in form of lipids, and in depth we assume that the reserve is in form of lipids in general.
For example, but it can be also.
It can be other in other ideas, and then you say these lipids can be converted back into energy. Yeah. At any time to carry out the body's physiological functions. Okay. Yeah. So you convert lipids into energy or energy into lipids. Where do we have mass energy conservation here? Where is the
In cellular?
In
cellular energy?
So do lipids have energy or?
No, they need energy to be converted.
They have energy and is converted into other types of energy. Yeah. Okay. So just to make clear, because sometimes it seems that you have something that is called energy, and then you convert it into lipids that is not energy, and then lipids back again into energy. But it's always energy, right? In different forms, you are using energy and in different situations. It's good to keep that in mind. Okay, so for me, I think it was very nice talking with you about this subject. I can pass the word.
Okay, thank you, Gonzalo. Now we're going to hear from Bastien.
Yes, thank you, Mr. President. So yeah, thank you first for inviting me. I'm really interested in those topics, so I was really happy to be able to read the PhD manuscript. Thank you for the presentation. So overall, I agree. There is a lot of work that has been done for sure. There is plenty of data. You have made very nice schemes and figures that I really appreciated. Sometimes they were a bit wrong, and Gonzalo showed us some of the errors that I also spotted. So you made some mistakes all over the, I mean, in terms of grammatic errors, but also sometimes you had sentences that were not finished. I feel that you were a bit in a rush at the end and didn't get the chance to read the final version. So I think you should really read carefully again and to remove all the sentences that are not finished, the empty brackets that are in there, and so on. So I have a list of most of them. So I will send it to you so that you can change that. Sometimes I thought also that you were going a bit too quickly to conclusions, and sometimes, for example, you say page 114, you say that fish need seasonality to improve their fitness, and this is shown by experiments that are doing research on hatcheries, for example, of fish in hatcheries, and when we put them back in the environment and they have seasonality, they don't have so good fitness. So this I think it's not totally correct. There's many other reasons to believe that it's not seasonality that affects the fitness of those fish. It's plenty of other different also parameters. And I was also thinking about what you said, page 140. You said this thesis demonstrates that DEB model is a good tool to predict the evolutionary potential of several traits. So when I read this, I thought, oh, that's maybe a bit too quick to the conclusions. It's a real question, but do you really look at and predict evolutionary potential in your PhD, do you think?
Not really evolutionary potential. But yes, like maybe in my mind it was more like fitness landscape than evolutionary potential. But I think with more further investigation, as I spoke about, like with genotype and influence, for example, and if we put some heritability in the model, yes, we can speak about evolutionary potential. So yeah, maybe it was too quick as a conclusion, but
yeah, I think so too. Yeah. I think you have a very interesting PhD, right? I don't question that. It's very interesting. You have nice results, but I don't think you can really speak about being able to predict evolutionary potential because you are missing indeed this very important part, which is the genotype effect and the capacity of the individual to transmit the trait, and because part of this variation between parameters might be environmentally driven, right? We maybe, I don't know, maybe there is a variability in those traits that are not genetic related. What do you think?
What am I thinking about to relate the genetic in?
Do you think it's only 100% genetic, those parameters? I don't know.
Okay. I assumed that in my work to explain like the individual variability across like the different values of the parameters. I don't think it's only genetics because I don't think there is like one genes or a couple of genes which can act to a certain value of the parameters, but I think that some genes can act on the values of few parameters, like for example to increase the value or to decrease the value, but not to a certain value.
It can be, yeah, it's
Can be like
pleiotropy.
really closely related, I think, but I'm not sure about that.
I don't know. I don't think that any people. I don't know if someone has clues here. Maybe Gonzalo. I don't know, or Dina. But yeah, we don't actually know. I think if it's only genetically driven, maybe it's also could be also a bit of environment of the parents through epigenetics or something like that. Maybe, I don't know. There are maybe some experiments out there where you can change the parameter based on differences in the treatment of the parents. Maybe have you seen some experiments on that?
Right now, don't remember any paper like that.
Mm.
But
Yeah, I think there are some some data out there where you change the temperature, for example, of the parents, and then you have differences in growth of the next generation, right? Even effects of contaminants and so on. So it's just to say that it might not be only genetically driven. So that's why I think you can't really go into saying that you predict the evolutionary potential. I was wondering, you don't speak much about the effect of the amount of food in the environment, and you always have F equal 1, right? So it's ad libitum food. Do you think that it would have changed a bit the results if you would have had F equal 0.5 or 0.8, which is often also used in nature? Probably the trouts they don't have always F equal 1. So how do you think it would change?
yes, in this work I focused on temperature. It was a choice.
Yeah.
But obviously, like if we change the value of F, it will change completely the results. And yeah, like F appears like really early in the schema of the depth, like at the assimilation flux. So if we reduce the F, like all the rest will be reduced, and thus the reserve. So it will change the capacity of expression of traits, but yeah, but maybe not the global trend.
Yeah.
Because
I think
the causality is already at the beginning of the process, you know. So
yeah, it's interesting. Yeah, I think the graph you have right here on that slide, PAM, actually it would be kind of a change in F as well, no? You would have exactly the same trend.
if I change F instead
of PAM.
amplitude,
PAM is having the same is involved exactly the same way as F, no? In the equations of that.
Yes, it's true, yes.
So I think part of the answer is already in there using PAM actually, where you can see that when you reduce F, then you increase the R. This is very strange to think that way. When you reduce the amount of food in the environment, you increase the fitness. I don't know. It's in my mind. It's counterintuitive actually, but I think it's true, right? You could see the effect of F using these graphs. It's very interesting, but you could also say that it's not exactly F that would change, but maybe PA, the amount of energy that is coming inside the animal. And this would change a bit indirectly it would be the F, but you could also say that in the environment, you have a given amount of energy, right? That is in the environment. That energy, if you increase the temperature, there is not so much reason to believe that energy, total energy, is also increasing. And this, you could also try to limit the effects of on your traits, life history traits, using this limit. That would be also one way to look at the data, and another way to think the data, I think. Yeah, so I'm coming back to what Luis Miguel said. In depth theory, you are able to have a thermal performance curve. This is something that we have a lot, actually, not a lot, but many species have that thermal performance curve where you have the increase and after that the decrease. It's pretty common. So I was wondering why you didn't choose that type of species rather than having species that were never optimized for those parameters.
Why I chose the species which what? I'm sorry. Like why I chose Daphnia magna or?
Yeah, if you look at the effect of temperature, I would go for species where you have the complete thermal performance curve, actually.
Actually, I start the PhD with Daphnia magna because it's like really well described in the depth theory in terms of parameter. It's really strong.
It's not strong for the thermal performance curve because it was never optimized for that.
Yeah, but for me, like just to model the deb at the beginning, it was and to compare like my theoretical traits, my quantified traits with real data, it was like relevant. So I continued on Daphnia magna, and after I changed to Salmotruta, and I didn't explore this thermal tolerance, and I thought that it's a limit. And yeah, maybe for further investigation, it could be like interesting, but at the beginning, just to model the just for the model, it was easy for me to have like a species like Daphnia magna which is really described in literature in terms of traits.
Okay. Regarding that temperature again, I was wondering, do you think that how is the amplitude of the seasonal variation for Salmotruta? Is it I don't know, is one of these amplitudes realistic in nature?
Not that much.
Okay.
Like it's really preliminary, like it's easily like sinusoidal, with like a high which could be related to the summer and the lowest point which is could be related to the winter. But it's not real data. It was just to see if there is like any trend. But I think like yes, after that we can use like real natural data for that. It was just more easy to interpret at my step of this thesis, but yeah, it's an exploration.
Yeah, no, of course it is. Yeah, I was just wondering if we are close to natural variability. I don't know in the Nivelle. I don't know. So maybe the 10 degrees is maybe close to what you can really see in nature. I was wondering, and so 10 degrees is not much actually. I think you could have gone a bit further even with the differences between winter and summer, which is probably a bit bigger even the amplitude. Yeah. And do you think that the periodicity is changing the results or not at all?
For me, periodicity can change a little bit, yes, because the expression of traits will differ between temperature. That we saw that with Daphnia, so I can assume that we saw that with, we can see that with Salmotruta, and with the context, like the current context, like we can have like change in periodicity, like in seasons. So yes, it could be interesting to vary this one too, or to just to model different climatic scenarios with like really high periodicity or low periodicity, which can relate to the current global changes.
Okay, and do you think that the mean level of the temperature also impacts?
The mean level.
I mean, you can have the
mean temperature at 8 degrees.
Yes. You could have the same amplitude, but having a different different mean temperature, right?
I think that yes, because we saw yes the really switch in Daphne Magna at a certain temperature. So maybe yes, it could happen for Salmotruta, but even if it's a mean temperature,
Yeah.
I assume.
Okay, and how can you mathematically explain those results related to the changes in amplitude?
How can I? What? Excuse me.
Can you think about a reason behind those effects that you see? Mathematically.
Mathematically?
Yeah.
I'm not sure I get it, but mathematically, if I have no amplitude, yes, it's a loss of seasonality, and like the A in my equation is equal to 0, and so there is still the ambient temperature, like fixed, and I'm not sure that you want.
Yeah, I think it's pretty clear what's happening with the amplitude. Actually, it's all driven by the exponential curve, because it means that you go above the mean, and so it's exponential, right? So it goes faster than the decrease, how it's happening in lower, right? Below the average. So I think it's all related to that exponential curve. It makes total sense actually that you see those effects. It's really driven by that. You would have a flat line, you would never have such an effect actually. So I think that's also interesting. It's all driven by this exponential. You could try to have a flat increase, a linear slope, linear increase, and you would never have the effect. Right?
Yes.
This could be also interesting to understand it a bit better. I was really surprised also by the variability that you see, indeed, what Gonzalo showed at the end. This variability that you see at the end of the graph of the increase there. I didn't write down the slides, but yeah, when you have the increase of intrinsic rates with the temperature.
Yes.
I think it's 42.
Yes.
Yes.
Same.
It seems that it's coming. It's very strange because it's the same animal, right? So it doesn't make sense to have variability around that. It looks like if you had different individuals and having variability in the parameters. So it doesn't make sense to me. It's probably related to numeric errors, no?
I'm not sure it's numeric errors, but just to be sure, like all the individual has the same values of parameters, and yeah, so for me it's just because of the value of temperature and not.
But since the temperature effect is following an exponential without variability around it, I mean, you should see also something that is following a general curve, I think. I don't know how you could check for that, if you have any numerical error behind that, but I don't know.
Did you look at the temporal aspect of it? Like, did it mean that these ones were fluctuating a lot over time?
Temporal effect of what?
This is all dynamic model you're dealing with, so you're averaging something that might not have reached any kind of stationarity. Perhaps it's just cycling a lot when you have strong effects of everything. So this is something that we didn't see much. We didn't see at all in your, I don't know how it works. You have these equations, you iterate them, but then you stop them at what points you record the trait, right?
Yeah, I record the traits, yeah. At the end, I record the quantity of energy in each energetic pulse, so...
But what's the end?
What's the end?
What do you say at the end?
At the end,
Because it's a dynamic model, right?
Yeah.
It's growing and things, and then you. Sorry, Bastien, to interrupt.
No, it's yeah, exactly what I meant.
Because there's some dynamic. It's called dynamic energy, right? So you have some differential equations that you integrate numerically.
Yeah.
And at one point, if you have some tiny, I don't know, differences, it might get amplified, and you have this type of issues that can come up. So I was wondering how you're doing it. If you did you check how it's running over time?
Yes.
And so you check that it's sort of stabilized before you record the value. You cannot do this for all these points, no?
It depends of which simulations, but sometimes I just take like the values at age at puberty. So.
And did you ever see something weird going on? Did you see them going to cycles, going to chaos? I don't know. I haven't worked with these modes. Can it happen?
It depends of what is your definition of chaos, but sometimes like there was like a lot production of eggs or something.
No, I'm talking about temporal aspects, like fluctuations.
Temporal aspects
that look erratic.
I didn't check individually each individual.
But the ones you checked.
What?
The ones you have checked, how did they look like? The one that
I didn't check individually. I already took like the value at when the individual was at puberty or the maximum value, or it depends on the simulation. And I analyzed that, but I didn't check individually each individual.
No, but in some case, did you check some of them or?
Yeah, but before implementing temperature.
Okay.
Like to see if the model fit with data.
I'm wondering if you change the time step of the ODE, if it
between Daphnia and Salle.
No. If you reduce the time step for Daphnia of the ODE, it probably can help actually to have something that will become that will follow something that makes sense. Reduce those variability around the curves. You should maybe look at that, because that might be a prompt to publish such a paper. I don't know how where it is now, but I think that might be something that people could say about that. I was unsure actually. I'm sorry, I'm going back to other parts of the thesis, but I was unsure how you chose the intervals actually of your parameters, because you said that for example PAM you limited it to 1,600. Why is that? You say in the PhD that you stopped where it stayed possible to make the species living, but actually PAM can be like super high, and it will not be a problem, I think.
You think it cannot be a problem that PAM is really high?
I don't see any problem related to that actually.
Yes, maybe a problem in. It's not a problem in terms of exploration, but like if I compare the highest value of PIM that I use for the magna, for example, it can be compared to like some values like the specific assimilation of big or huge carnivorous, like terrestrial carnivorous, which is, I don't think that Daphnia can have the same specific assimilation as this kind of species with not the same ecology and not the same food types. So it's for that for me some values are not really natural, but as it was an exploration, for me it doesn't pose any problem.
So why did you choose 1,600 then?
1,000 like individuals in.
No. The maximum value of I think it's.
So, okay. So values lower was where the model crashed, and the values higher after the highest range of values the model crashed too. So
it means that the model crashed when the PAM was above.
in my simulations, like my simulations crashed. So
I'm surprised of that actually. How high values of PAM can crash a model? I don't see really why. This is strange. Yeah, maybe you need to explain it a bit better in the manuscript because I was not very clear with that. So yeah, if we go a bit further and say it needs to be more realistic, more close to nature. It's what you said, right? You just said it's not natural to have very high values. Maybe do you think you could have done smaller differences, smaller variability within for the same parameter, and maybe you have information in the lab or somewhere about the variability that you could expect, like plus 20%, minus 20% around the mean, because I also felt that this huge range of variability between the parameters is a bit too high to be close to reality that what we could expect actually.
for me is, as I told earlier, so we need, if we want really natural parameters which can be observable, we have to focus on a specific population and re-estimate the parameters from these populations are. But from data we have in the lab, but it was not the aim of this thesis right now. For me, yeah, it was just to understand like how the biological processes can lead to traits. So
yeah, but maybe in the literature there are some information on those on the variability that you could expect in Daphnia magna for individuals that are reared in the same environment in terms of temperature and in terms of food. Have you seen something in the literature about that? I don't know. Huh?
About a range, a possible range.
At least in weight, for example.
I remember that I saw some range about the variability of the primary parameters. I'm not sure it was on Daphnia magna, but yeah, for me, we can re-estimate it, this kind of parameters, and yet to have like a specific range, it's not a problem. I didn't do that, but just because it was not my aim.
Okay. I have one final technical question and one broad question, if I have time. The graph with the heat maps, I agree with everything that was said. I was just wondering what it means to be at a intrinsic rate of 0%. I'm not really sure what this means actually. Is it no growth? Is it?
It is the lowest value recorded in the simulation of the intrinsic rate of increase. Yeah.
Okay, so it means that that's a bit strange though. So it means that the 50% is not half of the 100, right?
No. Yeah, no, it's not the middle. Not the mean, sorry.
It's yeah. So yeah, it makes it difficult to interpret, I think. Okay, so yeah, thank you. I have one final question. It's a bit related also to what was said before. But if you were able to get any type of data, what would you like to have to continue the work?
Me, I would like the data about like more genetic data, and so data and traits, and I'm really curious about like the link between genetics and bioenergetic processes. So yes, it's kind of the data I want to work more about that. Of course, the genetics and how it it is linked traits, but yes,
Okay, thank you.
thank you.
Okay, thank you, Bastien. I think now we've all grilled you enough with our questions. So now it's the time for the advisors to tell us a word about your PhD. So I don't know who wants to start.
Oui, alors je ne vais rien dire de scientifique, hein, ça je vais laisser ça à Charlotte, alors je vais te le dire en français, parce que mais même si je n'ai pas contribué à l'encadrement scientifique de ta thèse, j'ai quand même gardé un petit œil sur toi, et quand Stélia a soutenu il y a quelques semaines, je lui ai dit que c'était une vraie tornade dans le labo, mais il me semble que tu étais pas mal aussi dans le genre, en binôme, et tu es quelqu'un qui rit beaucoup, qui plaisante beaucoup. On t'entend de très loin quand tu ris. Et quand on te croise dans un couloir, tu as toujours l'air de bonne humeur. Je ne sais pas comment tu fais. Et tu as toujours cette immense banane avec les yeux écarquillés quand tu nous croises. Je voulais te dire que c'est tout aussi précieux que merveilleux parce que c'est contagieux. Et ça fait du bien. Et je voulais vraiment sincèrement te remercier d'avoir ensoleillé notre quotidien pendant 3 ans. Un grand merci.
Merci, je vais avoir la parole. Well, I might make it in English because I will speak a bit more about the project in itself, so that might also be of interest for some members of the jury that might not understand French as easily, but as often it will be less about science. And so this is also a first time for me, since this is my first time sitting here as a thesis supervisor. And I was glad that I did it with you for different reasons, and from both scientific and some not so scientific, maybe if Valérie already said them, and about all you brought during this project to the lab as a whole. But in terms of the project, what I really liked about how you embraced it is that you're very eager and very curious about what you're looking for. And that's precious. I think that's probably, well, we are both similar in some ways and very different in others. And that's some of the way we're similar. And I think that's something I felt really important, because whatever the hurdles, and there were some of them were methodological or technical, like scientific. Others were also maybe more personal at some times. Although you were still laughing, that hasn't always been easy. But you kept this motivation and this interest for what you were doing, and that made it very, very pleasant to work and discuss with you in this project. And you really had this wish to pursue different directions, sometimes too many at the same time, maybe. But it was I think something that you should keep for the rest, because that's important to keep this live and go on for this in research. And I think we both learned a lot, and me too, as a first-time supervisor. I learned a lot from you, both scientifically, but also on the more human side. And I think, you too, and probably something from what I feel, something you might have learned is how to combine this eagerness and maybe, as Bastien said, this wish to go on, and maybe a bit too fast sometimes, with the ability to sit down and think a bit more about what you have obtained. So think about the next direction and what they could be. And I think with this combination, you probably go on in a very good direction later on. And I think this project, so it started as a theoretical side to a broader project where, as Bastien said, we wanted to also get more genetic data, so things that would be more empirical on the basis of these parameters. And there was also the theoretical side that was to explore what the consequences of this genetic variation should be. And as quantitative genetics, especially for obtaining this type of parameters, they were a bit challenging to find. We started with the theory, as often, and I think this opened very interesting path. So some of them, yes, could relate to quite well-known patterns, more mathematical, but we also have some things that are more like emergent properties that we could pursue in the future. So that's very interesting. And I think both from a theoretical point of view, because yes, it opens new questions and ways to build different models. But I think also, as Bastien outlined, from an empirical point of view, it's also, as you said, now it's easier to focus on some parameters or some aspects of the model that might be more of interest to evolutionary biologists as a whole. So yeah, I would like to, well, first thank you for all this work and for having started also this line of research with me. And then, yeah, I think thanks also for, well, the rest of all you wrote, maybe more to the lab as a whole in this. And well, it'll be a good continuation on this path.
Okay, so on these words, I think I will have to ask everyone to exit the room, including Mélanie, so that we can deliberate. On va faire une petite pause peut-être. Yes, pause technique. So there's a technical break of 5 minutes. Right?
Sure, okay. See you soon.
C'est pas chaud.
Non, c'est pas chaud. Ouais. Ah, mais c'est une...